Welcome everybody to the Methylation and Brain section sponsored by Health Coach 7 and quite a few registrants. And of course, the big thing is listening to it when you're at your leisure, basically. So it is going to get posted. And as you can look at previous broadcasts on methylation, we've covered quite a few topics, starting off with putting bad genes on good behavior. So with that, I don't want to waste any time here. I want to get right into the subject matter, and I'm going to share my screen here so you can actually see it. Now, methylation in the brain is a very important topic because, as you know, you probably have seen on social media discussions about taking methyl donors and how these methyl donors may have helped or not helped in certain situations. And I think that one of the biggest concerns that we have right now as practitioners, one, because practitioners are getting confused as to how to appropriately address some of these gene variations or gene variants, they call them. And you know, when you are trying to address gene variants, it gets really tricky very quickly because of the fact that it may not necessarily, you can't read into the gene variants. It may not necessarily mean that the pathway in any way, shape, or form is actually affected in a negative fashion. So I have up on the screen here just a basic brain methylation focus. Okay, now I don't have the. You've probably seen methylation cycle with the whole cycle there, including the outliers in the amino acid and proteins and stuff like that. But what I basically did is I narrowed the focus down from looking at dietary intake and replenishment of certain nutrients and their influence on how it affects the bottom line, okay, for brain methylation. Now, what I really want to qualify here is that methylation, DNA methylation, is a normal process. It is a process by which DNA regenerates itself in a fashion that is consistent and constant. And then when we actually have any type of disturbances that are associated with the DNA's replication cycle, we often refer to them as either hypomethylating, which means undermethylation or the loss of methyl CPGs, influence on the methyl CPGs, or it means for hypermethylation, an increase in the methylating methyl movement of the cycle. So now a lot of people just think, well, isn't one worse than the other? Well, hypermethylation and hypomethylation both have their consequences, okay? And there's a lot of research out there that specifically will allude to that. If you look at a really good resource to look at here in methylation and understanding methylation is the Encyclopedia of Cancer. And the reason why I know as much as I do in epigenetics is because I've treated cancer for 20 years using immunogenetic strategies, and I've also treated autoimmunity the same way very successfully. Okay. And what I want you to see, this is the Encyclopedia of Cancer, very very pricey work. It's a few volumes, but I want you to understand. We're going to look at hypomethylation basically. Hypomethylation of DNA, according to the Cancer Encyclopedia, is a loss of methyl groups on the 5-methylcytosine nucleotide. Now, remember, when we talk about nucleotides, we're talking about single nucleotides, okay, and that's where that SNP word has derived from, single nucleotide polymorphisms, because the methylating cycle is affecting the nucleotides, okay? Now, it's important for you to know that different mammals will methylate differently. For instance, what might be hypomethylation in a human might be hypermethylation in a chimpanzee. Okay, so you're going to find that the different species in genome will have different methylation rates, and those methylation rates may not be indicative of any kind of a disease process at all. Okay. Now, bottom line is we're talking about brain and methylation here, and we're looking at hypomethylation. And as you can see here, hypomethylation can be applied to unmethylated state of CPG sites, which are the little islands that are influenced by the methylation. And the characteristics of these issues of methylation could be found in several different types of disease process, not just one type of disease process. Now, as you can see here, we're seeing that as this went on, characteristics would be seen that global hypomethylation, which means a systemic global hypomethylation, is typical in aging cells. So you're going to actually see it associated with cells that have premature aging. Okay, you're going to see hypomethylation as well as hypermethylation in tumor formation. Okay, so you're going to see it in cancers. You're going to see hypomethylation in certain types of blood disorders, immunodeficiency disorders. You're going to see it in different types of anemias. Okay, you'll see hypomethylation influencing chromosomal integrity, because the integrity of the chromosome depends upon a reliable methylation cycle. So as you can see, hypomethylation, that's the undermethylation or low methylation, if you will. And as you go along here, we're saying, well, what does methylation do? I mean, what is it? What does it entail? Basically, we're seeing the methionine circle. We're seeing the THF, which is the methylene tetrahydrofolate circle. We're seeing the glutathione circle. We're seeing the neurotransmitter relationships right here. Okay, as you can see, dopamine, some of the catecholamines like norepinephrine and epinephrine's part of that, then serotonin and tryptophan. Okay, so you can see that if you enlarge it a little bit, just look at it a little bit closer. I'm sorry for the blur, but here's where you see the CBS cycle. Here's where the blood-brain barrier crossover is. Here's where you see BH4 and BH2. So some of these you recognize. Okay, here's where you see the free radical influences on creating more disturbances in methylation cycle in the brain. I mean, so hypo and hypermethylation both can affect the brain in a negative fashion. Now, here's a very interesting site that I want you to look at. It says, what is epigenetics? Bottom line is, when we talk about methylating cycles, whether we want a normal DNA methylation cycle, I know that this is very complicated to understand because it's using a lot of biochemical terminology, but in simple, when you're talking about DNA methyltransferases, you're basically talking about an enzyme that's going to transfer the methyl groups from one place to another. And DNMTs are the main source of the enzymes that are required to establish the good maintenance part of DNA methylation patterns. So now if you see methylation cycle being altered, then you're going to see DNMTs, 1, 3As, Bs, all involved in this process of disturbance in methylation. Now, we all know that the end result of methylation disturbances can result in very severe type of disease process like cancer. Okay, if you actually highlight hypermethylation is an increase in normal methylation levels, and then if you looked at hypomethylation, which is down here, it's going to tell you that it's a decrease in normal methylation cycles. Okay, just to kind of give you a little bit of an overview of this methylation movement. Now, it's very important for us to be sure that we are looking at methylation from the standpoint of gene controls. Now, here's another interesting study here, which was published in 2010, which I found to be very, very informative. Now, you want to be skimming through the research, and a lot of this stuff here you might not understand. But bottom line is that we never ever should be basing our treatment just on SNP variations. What we really need to be doing is we need to be looking if we have disturbances in methylation, and we know that the brain is very sensitive to those disturbances. We need to be looking at causative factors and the peripheral factors that are causing the disturbance in the first place in the DNA, okay? And then address those, because if you actually try to fix the methylation cycle before you address any of the environmental factors around it, you're going to have a problem. And that problem could be exacerbation of certain symptoms, because what you wind up doing is provoking provoking the bear, basically. Okay. So if you've got something where your body is trying to detoxify, for instance, heavy metals like mercury, which is very toxic to the brain, or any of these other toxic sulfur agents or xenobiotics, here you go trying to repair the methylation cycle when the brain is already struggling to stay balanced. And the methylation cycle is already disturbed one way or another. And in a lot of cases, it's hard to tell whether or not it's methylating, hypermethylating, or hypomethylating. Usually, inflammatory processes are associated with more hypermethylating movement, but not always, because you can actually see hypomethylation in inflammatory movement as well. And the only real adequate way to be able to try to figure out which cycle is happening in the brain is by looking at specific gene variations. Now, see, I in gene cards looked up hypomethylation in brain, and as you can see, there are 227 different gene variations here that actually are involved in hypomethylating movement in the brain. And you can actually export this list out and do a study on most of it. But you're going to see a lot of different things that are recognizable. You're going to see MTHFR, which is recognizable, and you can actually profile the MTHFR by looking at more publications. And as you can see, there are a ton of publications there that are actually demonstrating hypomethylating relationships in certain type of cells of the body, including anything that's going to be involving neuronal tissue. Okay. You also could see that DRD1, which is one of the classes on dopamine receptor. Of course, you've seen DRD2, DRD4, DRD3. Very important in looking at this because of the fact that, when we actually try to profile it, you can see that there are certain type of risk promoters in dopamine. You can actually see the BMP4s, the COMTs, which you probably recognize. So when we're talking about brain methylation, right? And you're looking at your conversion report and you see all these different gene variants, and then you go for the gusto and saying, I'm going to take, MTHF, or I'm going to take something that's going to energize my liver, or I'm going to take this or that, you wind up creating more harm than good, because if we haven't addressed the environmental factors surrounding the issue of the disturbance, we have a bigger problem to deal with then. So it's important to stay focused on priority and triaging how we want to approach it. So again, you can see tumor necrosis factor is actually a tumor formation oncogene. HTR2A, you've probably seen that on your genetic reports, would actually involve serotonin. I mean, there are all of these different ones here that you see. GAD1 is a very famous one for breaking down glutamic acid in the brain. And the thing about it is that you're going to find that people that struggle with a lot of brain methylation cycle disturbances will have more expression at, double mutation, or what they call a positive or a heterozygous relationship with GAD1. So it's really important for you to know that the focus has to be on identifying the environmental factors surrounding and then trying to address those so you don't create a bigger problem. Okay? A very interesting relationship there. Okay. Now, I found that this was an interesting study, and you may want to probably look at this because this is, demonstrating DNA methylation in humans, and how it actually begins, and what it actually involves, and then it compares it to the disease process in general, or cancer specifically, breast cancer, even more identifying detailed cancer. And you're going to find that you'll learn a lot with the relationships of methylation here, because you're trying to figure out, okay, how can I How can I actually impact my epigenetics? And the bottom line is what we're talking about is epigenetic programming. So if we want to change certain scenarios in the brain where we're actually having a lot of neuroadaptive disturbances, what that simply means is neurotransmitters cannot do their job appropriately because the methylation cycle is so off because there's some provocative factor that's affecting it, and it's causing a real irritation to the expression of cellular DNA. And remember, cellular DNA is actually expressed in your neurons. Now, your neurons are very, very sensitive. Neurons are probably the most sensitive cell of the human body. Okay, you have a lot of forgiving cells, like your liver cells are forgiving; they can take a beating. The intestinal cells, man, they are just really doing. They're diversified. They got many hats. Okay, but your brain cells, they got mainly high specialized movement in trying to create signals and reliable signals to actually program an intensified network to try to keep things balanced all the time. And you got to remember that those cells are very, very sensitive. Okay? Very, very sensitive. Here's a very interesting study right here. You need to really read this study because this study is actually talking about the GWAS DNA methylation analysis of the human brain in schizophrenia patients. It's a recent study of translational psychiatry. I found it very, very interesting because when you look at it, bottom line is you're actually starting to see various types of deficiencies associated with methylation impairment. Okay, and there's a bunch of genes that are named here. You actually might recognize some of them, like the HTR2A and the COMT, right? You can see those. It's DRD2 is also part of that process, okay? But if we actually go down and we try to, some of this is going to be hard to understand, of course. You don't need to understand it, but just go to the summary and discussion, and it's probably going to give you a good handle on what the methylation movement is in the correlation with schizophrenia, okay, and how it's expressed, and what the conclusions are. And they're finding more and more that higher global DNA methylation in patients with schizophrenia, as it says right here, okay, are found to be, what they're saying is the main limitation of our study was that patients were not free of antipsychotic medication. So what they're trying to find out is how antipsychotic medication affects the global DNA methylation expression, and they're finding that these drugs were not really associated with changes in methylation, which is an amazing discovery right there, because what is that telling you? Are these antipsychotic drugs really working appropriately? Are they creating more complications or more layering? And maybe there's a better way that we should be approaching this with, looking at other neurochemical movement by using specific type of neurotransmitter receptor initiators. Figure out some other way, right? This is a very difficult disease to treat, but DNA methylation signatures on schizophrenia is a very wide field of study. Here's another study. This was actually on the impact of athletic injuries to the brain, to the head, and the impact of the concussions on epigenetics. And they're talking about, hypomethylation here, showing a decrease in normal methylation levels, and that following injury, there was pretty much on average, a global hypomethylation observed 4 days after the injury. Okay, which shows you that inflammation not only can cause hypermethylation, but inflammation can also cause expressed hypomethylation. All right. And the whole purpose of this article was basically to show epigenetic process mediated gene responses to change in tissue environment, and over time can confer memory with whether a gene is expressed or silenced. And you can see that post-concussion or brain injuries related to various type of impact sports, how it can affect the methylation movement of the brain and contribute to functional disturbances of memory and brain function as a whole. Very, very interesting study there. Okay. All right, here's another one that's talking about, this is a very interesting study in comparison to humans with chimpanzees and the correlation between how hypomethylating patterns could be hypermethylating patterns in humans, and how the genome, because of the genome differences, can manifest with different methylating cycles in different species. Okay, they start off here, really interesting, saying that DNA methylation is a epigenetic DNA modification, and it is, okay. DNA methylation is a crucial epigenetic process, and you have to understand that if you want to influence epigenetics, then you do have to address methylation balance. But you have to do it by focusing in on the environmental factors that affect it systemically. So you have gut problems, got to address those. If you have liver detoxification problems, exposure, antigen processing problems, if you actually have heavy metal toxicity, if you've got continuous overstress in your life to where you're always in a fight, flight, fright mode, which profoundly affects the methylation cycle, by the way, either way, hyper or hypo. If you've got any of these things that are happening, if you've got a poor diet that's filled with all kinds of processed junk, if you're living in an area where there's a lot of particulate matter in the air that's dangerous, if you're living in a house that has a lot of mold and fungus, if you've got situations that are adverse, you got to try to help the body to adapt to these things more favorably to help balance out the methylation rhythm. Okay, now hypermethylation has about 567 genes associated with it. So you're going to be looking at and saying hypermethylated in cancer, HIC1, which is actually a known gene variant. You've got, EGFR, which is epidermal growth factor receptor. You've got several ATP binding cassette proteins. If you keep on looking all the way down the line, you're going to see, if you actually go to export the list, say I'm going to export to, just show symbols. There you go. There's your symbol of every gene that's there. And then the thing about it is that you can identify some of these genes on your conversion list, okay, and your conversion report that you've actually generated. And when you start looking at these, you don't want to start freaking out and saying, "Oh my goodness!" Not all of these are negative influences on methylation, but I want to point one out here. Here's one that's brain oriented. You've got GSTM3, which is a glutathione S transferase muprotein 3, which actually simply means that it's brain derived, and it's very important in what they call maintaining detoxification of toxic compounds as it affects the brain. As you can see, it's involved in conjugation of reduced glutathione of wide number of compounds in xenobiotics that actually can influence the blood-brain barrier, cross it over. So that's where glutathione is very important, and that glutathione rhythm assisting with healthy methylation balance, because anything that's going to affect the brain in a negative fashion that's toxic, this particular S transferase glutathione actually is important in sustainability. That means long-term sustainability of detoxification in the brain. And you're often going to see, more articles that'll actually go into the importance of polymorphisms that are associated with that. But I wanted to just point this out to you because these are one of the genes that come up, gene variations that come up consistently in a lot of people. This glutathione S transferase mu protein that is really, really essential in assisting with detoxification of toxic compounds. So now you can do more research as you go along there, but I think that you're getting the point. Here's a very interesting book, and I've read segments of this book. You can take it out in the library. It's called The Developing Genome, An Introduction to Behavioral Epigenetics. And I think that you would find that he tries to speak in plain languages, and he's looking at different species across the board. But I think that this would actually give you a feel for how epigenetic expressions are and adaptations manifest in different species, but he spends a lot of time talking about humans in here as well and the correlations between them. So I think that you'll find that that's interesting to at least maybe look at. Um, then this specifically, this is a very complicated one. This is from Genome Biology. I came across this article when I was at a conference in the Genome conference in Boston, and it's actually talking about methylome identities, which simply means that the methylome are the genes that control DNA methylation. So methylome simply means specific genes that control methylation movement. And this here is talking about the human brain methylome and tissue-specific epigenetic variations across the blood-brain barrier. So I think that you'll find that this is interesting because it names a couple of different genes that we often see, including the BDNF, which is the brain-derived the neurotrophic factor, BDNF. And you're going to find that that's a very strong protective gene variation. So if you remember, when you're actually going to gene cards and you're going to be looking up a specific gene, you can just type it up in the search window, BDNF, and it's going to pull it up as brain-derived neurotrophic factor. And then you're going to look at it and profile it and see, well, what does it really do? How does it really protect? Well, this gene may play a role in the regulation of stress response and the biology of mood disorders. So it's a very strong brain-derived gene control. You can do more reading on it yourself. But methylation is definitely, that's considered a brain methylating gene. Okay? And a lot of people come up with it double positive, which is red and red-yellow combinations. Okay? So anyway, I think that you're getting the point here of how many real complicated things that we, try to simplify that we really can't simplify for the sake of, just trying to, say, okay, well, if I could just focus on methylation, I'd be fine. Well, I'd say not really. You got to be focusing in on the environmental factors surrounding it to assist with the body's ability to address what provocative factors are causing this methylation disturbance in the first place. So I really encourage you to be looking at some of these different articles and such will be available. You can actually read on your own. But bottom line is that if you're going to work with methylation, and my word of caution would be this: any practitioner that you work with, you want to be sure that that practitioner is aware of your total health picture. You don't want to be just having the doctor just give you this and give you that. And make sure that you're working with a team that is actually going to coordinate the best movement of effort. And the thing is that you can't start addressing everything at once. You're going to have to chip away at it maybe a section at a time and realize that it's essential because these little minor frequency changes go a long way. And it might take you maybe 9 months to a year. And if you're trying to rush something, you're going to be very disappointed because it doesn't happen that way. The thing is that these disturbances exist over a prolonged period of time. And then all of a sudden, in an instant, when you're aware, this might have been a cycle disturbance that have been going on for the last decade in your life. And so all of a sudden, because you become aware of it, you can't fix it in an instant. You're going to have to work at it a section at a time to whittle away at it for your health's sake. That's the whole inspiration of this whole process, is that you can actually look at this and you can begin to create the repair strategy for it. Okay? And remember, little changes for the brain go a long way because those neurons are very, very sensitive. So it's not like, oh, I got problems with serotonin, I'm going to take tryptophan. I load my brain up with tryptophan. All of a sudden, I got a flood of tryptophan in the brain. And maybe you do well for a month, but then all of a sudden, boom, something happens. Okay? No, you don't want to be loading up on single nutrients and flooding the system and trying to accomplish. You want to do this step by step. Try to identify environmental factors first that are surrounding the issues. Gently work with the brain, very gently work with the brain. The brain is a delicate structure. It responds to small changes. It doesn't respond to drastic flooding and superdosing of anything. So I really caution you to be strategic about that. So anyway, I leave it open to questions here. Wanted to give you more information here. I wanted to say, everybody, there's no such thing as a dumb question, because not in genetics, because there's so much information out there. Okay, I got one question. Okay, well, how does SAMe affect my body's ability to use things like MTHF or folic acid? Well, I don't like to use the term folic acid. Folic acid is a synthetic vitamin, and it really you really want to be looking at it from the standpoint of biochemical compatibility. Calcium folinate is actually an accepted form biochemically. Folinic acid is an acceptable form. Methylated quatrefolate is an accepted biochemical form. Now, how is the SAMe affecting COMT pathways, and how does it affect bottom line methylating process of the folate? Well, it can cause more accelerated symptom effects, and it also has some influence on dopamine receptor. Now, SAMe is a great product, and it actually can help with prefrontal balance of dopamine, and it can actually assist with better homocysteine metabolism. However, if a person is having a lot of CBS activity and a lot of BHMT disturbances with the gene variations, you might find that SAMe, starting off with it at first, may not necessarily be a good strategy. You might want to just ease into just doing very, very light replacement by maybe taking the back alley approach by doing, cofactors like P5P, assisting with maybe movement of uh, the pathways to accelerate more, I would say, homocysteine balance. Some of the things that actually come into play there are folate transporters, which are known as biotin, uh, thiamine, very important cofactors in assisting with folate movement and taking the load off of the methylation cycle. And then once you see that you're getting a pretty reliable pattern of, no crisis reactions or aggravating reactions, then you can begin the process of, adding a few more things in there. So now, when you're looking at this, B2 is important, P5P, and also I don't like pyridoxine. It's just an inactive form of the vitamin. I don't like folic acid. I would say calcium folinate's the form, and I don't like cyanocobalamin because that's a very synthetic. It's synthetic garbage, basically. Methylated B12s are great, but sometimes people are very sensitive to that. And if you're having a problem with the BHMT and COMT sensitivities, you might find that methyl B12 to start out with may not necessarily be your baby. Okay, you may find that it creates more aggravation for you. So you just have to be strategic. It doesn't mean forever. It just means, gentle. Then some people start out with very high doses of glutathione, thinking that they're going to pump up the cycle, really affect this detoxification. And what they do is they overdose on glutathione right off the bat. Not a good idea, people. I mean, it's like, don't be super dosing your glutathione and trying to energize your detoxification to the point where you create a dumping syndrome, which you actually start dumping all this crap into your bloodstream, and your liver then goes nutso, and then your brain is going to not be happy. Okay, so you got to do things strategically, not all in one type of deal. Glutathione, you start off very low, slow drip, just let your body get used to it and go from there. Now, another question is, if a person has Epstein-Barr virus, and if they're experiencing, more symptoms of, associated with the virus, like fever, is it possible that, uh, you know, let's see, if I'm taking L-lysine every day, and if I sweat even a few drops, I have fever all night long. I sweat very easily, so I'm not sure if it's methylation issues or something else. Well, that's a good question, because a lot of the times what happens there is that the methylation disturbances are there for sure with viral infection. And if your body is actually has an autoreaction, an autoreactive immunity, say for instance your body's mounted an immune response against the virus, whether it's Epstein-Barr or cytomegalovirus or the HHV6 stealth viruses or any other virus, the problem is methylation disturbances are created by an I would say, responsive immune system, to where then the immune system is autoreactive against itself. And yes, the methylation issues can create these disturbances. And the other thing is that if you focus in on trying to fix the methylation, it can create more of an enabling on the autoimmunity. So what you really have to do is you have to identify where the flaws are in the immune adaptation, and try to address and calm that autoreactivity down, because with that autoreactivity comes higher risk for inflammatory destruction of healthy glands. So in other words, that sweating could be a sign that the antibodies are hitting your adrenals and your pituitary, or it could mean that you're secreting higher amounts of T cells and cytokines in response to what the body thinks might be still infection on board. So the key is, are we still infected? Do we still have active virus on board? Or are we post-reactive? Are there other cross-reacting viruses or exposures that I might be exposed to here? And I'm just thinking that it's the Epstein-Barr, because it's something else. So those are the things I'd be looking at. Okay, very important to look at those. And okay, so if one person has a high sulfur levels, should we indicate our medical records we are intolerant? Possibly. I would say that would probably be a smart thing to do. You may not be allergic, but you could be intolerant. So it's very possible. Very good question. Very, very good question. Anyway, I wanted to thank everybody. I'll have these articles present. You'll be able to listen to this again, Methylation in the Brain. Look forward to our next session that's coming up next week. You can go on the website and see what the topic is for next week. And we're going to be talking about methylation for a little bit now, all the way through September, just to cover it more thoroughly. Okay, and I think that we will be you'll be a well-versed bunch of people on methylation. There's one last question. How do you like NAC? Well, I think NAC is a great antioxidant, and I think it is a very good detoxification nutrient. But I also believe that you probably have to do it with caution, and not try to, one of the things that you want to do is make sure that you're looking at the whole plan. And that's something that we're going to be definitely doing. That's what I do with all my clients, is when we get them on a plan, we get them on a balanced plan. And they're not going to be getting a nutrient that might be just too overpowering for them. So I think that's the key at this point, is to be sure that we've got that balanced plan. All right, thank you, everybody. I appreciate your attentiveness. Look forward to this recording, and we'll see you again soon. Bye-bye.